So I guess you're seeing my screen now, right?
Yes, we
Is
can see
that
your
so?
screen.
Okay,
Yes, yes.
good.
Perfect. Okay, we'll wait
Um,
for 2 more minutes. In the meanwhile, I'm going to introduce Dr. Mignon. So Coralie has worked in the Interdisciplinary Center for Smell and Taste at the University ENT Clinic in Dresden, where I work as well. She studied in France before this, where she got a bachelor degree in biochemistry, cell biology, and physiology, and then a master's degree in physiology, neurosciences, behavior, and cognitive sciences. Then she did her PhD from 2016 to 2019, when I was just in college still, in neurosciences at Strasbourg University in France at the iCube laboratory and worked on the modulation of cerebral activity by subliminal odors. Currently, she is focusing on the central nervous processing of olfactory information in healthy controls and patients with migraine. So I think, honestly speaking, she is the person who knows the most about neuroimaging tools. And that is exactly why I thought that she would be great to talk to all the medical students about it. And on the methodological side, she uses psychophysics, EEG analysis, and MRI-based techniques. And last but not the least, she's also a member of the GCCR, quite similar to GCMER, which is the Global Consortium for Chemosensory Research. And this consortium, actually, the interesting thing is that they had a study, or they have an ongoing study in which they are investigating smell at an international level, both smell and taste. And they were actually one of the first people who brought about the diagnosis that anosmia is one of the first symptoms during COVID. So I think that we quite underestimate the importance of smell and taste, both being higher neurological functions. And I think I can tell you that today's webinar will be very useful for anybody who is even slightly inclined towards neurology, neurosciences, or neuroimaging, or even behavioral sciences, because assessing behavioral sciences also requires neuroimaging. So since it is 4:05 already, and 7:35 in India, without further ado, I would like to ask Dr. Mignon to please start the presentation, and any questions that you have, because some of it might be a little different from what we have learned so far in our MBBS. So if you have any questions, feel free to write them down in the chat, and I will stop Coralie at the end of the EEG section, before the MRI section, so that we can discuss all the EEG questions, and then we will go on to the MRI section. Is that okay?
for me, that's perfect. Thank you.
Perfect, no problem. Okay, so I think we can start now. Go on, Dr. Mignon.
Thank you very much. So I'm very glad to present this lecture today. And as thank you for the introduction, that was very good. I feel a bit old now that you said that you were in college when I was a PhD, but that's okay. So I'm gonna talk, as you said, about EEG and fMRI, and it will not be tons of examples about research with EEG and fMRI, but more about the techniques themselves. So the aim of today is that when you leave this presentation, you have knowledge about what neurologic processes are behind these two techniques, and you have an idea also on the steps required to get results with these techniques. So today is mainly for very early beginners with these techniques. If you want more information, I can answer questions, but please refer to some other courses because it will be very simplified. So let's start with EEG first. So what is EEG? EEG is measured with electrodes that are placed on the scalp. So if you look at the picture on the left, you see there are 64 electrodes, but of course you can have more or less depending on your research question. And these electrodes, as I said, they should be placed the closest possible to the scalp. So usually we use some gel to improve the conductivity of the signal. And the electrodes, they measure the postsynaptic potentials of pyramidal cells. So you probably know in the cortex that there are several layers, and the layer that interests us is the one with pyramidal cells, because it's these neurons that are perpendicular to the scalp, and it's the only way with these electrodes to measure the signals that are perpendicular to the scalp, which is not the same with the MEG, magnetoencephalography, for example. But for EEG, we measure only these postsynaptic potentials of pyramidal cells. If you look at this figure on the right here, you see that you can have a signal with negative deflections or positive inflections. And by convention in EEG, the negative potentials are up and the positive potentials are down. I don't really know why, but I guess it's because it's reflecting what happens in the pyramidal cells. So I'm gonna explain that to you now. And when you measure a negative deflection, it's because the gradient inside the pyramidal cells is from the bottom to the top. When you measure a positive potential, it's because the gradient inside the pyramidal cells is from the top to the bottom. And this is depending on inhibitory or excitatory cells and on the placement on these cells along the axons of the pyramidal cells. So depending on that, the gradient is different and the signal that you record with the electrode is different. So the signal itself, it's a voltage, so it's expressed in volts or microvolts along a period of time, so across time. So when we place the electrodes, we want to be sure that we can understand which signal comes from which area on the scalp, and we want to be sure that we can reproduce it from one subject to another. That's why when you use EEG, there's an international placement standard. So the most famous and used is the 1020 system. And with this system, you have a standard of naming the electrodes. So if you look at the left figure, you see that the electrodes have all a letter at the beginning. This letter corresponds to the lobe that is below the electrode. So F for frontal, C for central, P for parietal, and so on. Then you can have a number or a letter. So if you have Z, it means that it's on the central line. So Z is for the line that goes from the Nasion to the Inian. If you have a number, so the odd numbers are on the left side and the even numbers are on the right side. And then you go from the middle to the external part of the head. So that's how the placement is. So if you read some articles and you see that they talk about CZ, PZ, or P4, you know obviously where it was placed on the scalp. Okay, so now I would like to explain to you that there are two kinds of EEG. So you can record continuous EEG or you can record event-related potentials. So continuous EEG is when you study the brain waves among a long period of time. So you can have some stimulations at the same time, or you can ask your participant to do a cognitive task at the same time or nothing. But for event-related potentials, it's an automatic response of the brain to a stimulation. So you always have a quick stimulation for this kind of experiment. So let's see the continuous EEG first. So if you look at the left panel here, So what you have to understand is that the signal in EEG, it's a mixture of subsignals. And these subsignals can be represented by sinusoids with different frequencies. So there are different frequency bands in the EEG signal, delta, theta, alpha, beta, gamma. And these different frequency bands, they have different frequencies. So you can have low frequencies. This is the case for delta, for example, 0.5 to 4 hertz. And a low frequency corresponds to a very, very slow signal, as you see here. And you can have high frequencies. So it's the case for gamma, 32 to 100 hertz. It means that the signal is very fast. So the aim when you do continuous EEG is to do what is called a time frequency analysis. And the time frequency analysis allows you to know what is the amount of each of the frequency band in your overall signal. And why is it interesting to know that? It's interesting because some cognitive processes are known to be mainly from one type of frequency band than the other. For example, if you're resting, if you're just relaxing, and then you're closing your eyes and the EEG is recorded, then in the EEG you should have mainly alpha bands. So you will see that your signal is composed mainly by alpha frequencies and not a lot of theta or delta or beta or gamma. So the next, I'm going to talk about event-related potentials, and I will stick to that until the end of the presentation about EEG. So as I said, event-related potential allows you to see the automatic response of the brain to a stimulation. So one example is here on the right. So you can send any kind of stimulation. It can be visual, auditory, gustatory, olfactory, mechanical, any kind of stimulation, but it has to be very quick because you need to see the automatic response of the brain. And when you send a stimulation, you will see the response very fastly, so very fast after the stimulation. And these responses, they are known for their shape, and they are more or less locked in timing. So that's what we call latency. So depending on the type of stimulation, you know for healthy subjects where it should happen, when it should happen. And they are more or less locked also in duration and in amplitude. But there are inter-individual differences depending on the health status of the subject and also from one individual to another. Sometimes the amplitude or the latency can be a bit different. So it's interesting to use that because, for example, if you want to compare a control group of subjects, of healthy subjects to a patient group, you can measure the latency and the amplitude of your signal and see if it's different depending on the groups. So that's an example here of a study that we are doing in the lab. So I just gave you the signal for the PZ electrode, so parietal, and it's the signal for one subject who received CO2 in one nostril. So CO2 is the gas that you have, you know, in Coca-Cola, for example, and here it was sent in the nostril, so it's a bit painful. And we see that at time 0 here, the stimulation was sent, and around 0.5 seconds after, there was the response of the brain here that is very clear. And we can see typical peaks in the signal. So in this response, P1 for P for positive, N1 for negative, and P2 again positive. And these peaks, we can measure them for one individual to another and compare them between some groups. So in my lab, what we are going to do is that we compare the CO2 response between healthy subjects and migraine patients, because CO2 is painful, and obviously in migraine patients, the pain processing is a bit different from healthy subjects. So that could be translated into a difference of latency and a difference in amplitude. So overall, when you choose one type of stimulation for your experiment, you have to be very precise and you have to make sure that none of the other sensory systems can be stimulated at the same time. Because if you stimulate different sensory areas, then it's deep in the brain, and the EEG makes no difference between the location where it arises. So on the scalp, you will have a response, and if the two responses happen at the same time, you can't know on the EEG which was due to, for example, auditory or due to olfactory. I just give you an example here. In one study, in this study actually that we are doing, we send the stimulations, the CO2, with a machine that is called olfactometer. Each time there's a CO2 stimulation that is sent, the machine, the device, does a sound, like a click. And we need that the subject doesn't hear the click, because if he hears the click, then we can't see if the response is due to the noise, to the click, or to the CO2 stimulation. So that's why, to avoid that, they have to wear some headphones, and they hear some white noise at the same time, so they don't have this quick stimulation from auditory processing. Yes. So then when you have when you send your stimulations, you cannot send just one time your stimulation. You have to repeat it a lot of times, because you need to average your responses together. Why you need to average? Because if you look at this figure here, there's a lot of noise in the EEG data. Here in the end, you have the overall signal with the noise and the signal of interest. Here in the middle, you have the signal of interest without the noise, and on the left, you have the noise alone. So what you see is that the signal and the noise together looks really similar to the noise alone. But when you average all your stimulations together, then you see that you're able to have the response to the stimulus of interest. So that's why you need to average your stimulation, because you get rid of the noise and you keep only the stimulations. So it means that the more stimulations you have, the better the final signal is. But the longer experiment is, the worse for the signal it is. Because the longer the experiment is, then the patient becomes bored or tired, and then the quality of the signal is not as good. And also the more demanding in computer space and data analysis it is, because EEG data can be very demanding in computer space. So now I'm just gonna give you some example of artifacts in EEG that we can see frequently. So you understand that it needs to be filtered at some point, because there are so many possibilities of artifacts. So the first and the most common one is eye blinks. So that's what you see here, the very huge amplitudes in the signal. When people blink, it's negative deflection. There's also artifacts caused by eye horizontal movement here. Lead movement, when the participant pulls on the wires or moves. Also muscle artifacts, so it's usually these very high frequency bands here in the signal. Instrumental or environmental artifacts, usually at 50 Hertz, so it's what you see here. For example, if you have another device, electrical device, that is plugged next to the EEG amplifier, you can have such a noise in your signal. Or ECG, if you look at this line here, you see that there are very high amplitude peaks at a very regular rate. So it's ECG. Or BCG, here it's everywhere. Sweat also, the sweat can lead to a distorted signal. Here you see the line is not straight anymore. And if there's a phone next to the amplifier and the phone is ringing during the experiment, then you can have very high artifacts in the signal too. So overall, you should ask your participant not to blink too much during the experiment, not to move, and to switch off his phone before the experiment, to try to avoid as much as possible the noise. So after that, when you get the data, I'm not going to explain the different steps because we have to talk about fMRI too. But after that, when you get the data, you also have to filter them, because it's not because your participant didn't blink, didn't move, that you don't have any noise. There's lots of noise in the EEG signal. So you have different pre-processing steps that allows you to get rid of the noise. And then in the end, you should average it to have, as I showed you an example with CO2 before, to have the event-related potential. That was all for EEG. Thank you. So if you have any questions for this first part, then feel free to ask.
Yes, so we actually have a question which was asked on private in the chat. So the student is asking, how do you identify the eye movement artifacts?
So there are different possibilities. I will go back to this if I can. It doesn't move, okay. So the way you can see it, it's like when you're used to it, it's very easy because the amplitude is so high that you know that it's an eye blink. And also you can put some electrodes, extra electrodes on the face to be sure. So you put an extra electrode above the eye, the eyebrow, and you are sure in this way that when you see, it was here. Yeah, when you see, so usually it's called FP2. FP2 is an electrode that is usually placed above the eyebrow on the right side. And when you see that the amplitude is very, very high on this electrode, like here, you see that it's an eye blink. It cannot be an event-related potential because, as I said, you need to average to see a response. You can't see usually a single response just on the EEG like this. So here it's an eye blink for sure. And you see also that it's reflected mainly on frontal electrodes. And then when you go to temporal or parietal electrodes, it's not there anymore. So for sure, it's an eye blink.
Okay, understood. We have one more question.
Yes.
The question is, what do you do in your lab? How do you identify migraine EEGs?
So I can explain a bit more the study with migraine patients, but I'm not sure I understood the question about identifying the EEGs from migraine patients.
I think they're asking that, is there like a particular pattern that you see in migraine, or like for example, if you're presented like an EEG, then
Yes.
can you, if you know where the events were, like where the stimuli were given, so can you look at an EEG and say that, okay, this is a migraine patient's EEG?
No.
Yeah, okay. So
I don't think so. It's not as typical as an epilepsy, for example. But for sure, some of their brain processing, especially the pain matrix, are impaired. So if you find the right procedure and you compare them to healthy subjects, for sure you will find some differences, but it's not a marker of migraine. It could be also an impairment for another kind of disease, I think.
Yeah.
And what we are doing in the lab for this is that, so we send some stimulations of, actually, at least in the project I'm working on, we send, and also you are working on the same project with children, actually. So we sent some olfactory stimulations and CO2 stimulations to the patients and to the control subjects. We record the EEG. We do event-related potential, so we average olfactory stimulations together and CO2 stimulations together. And then we compare their peaks, you know, the peaks on the EEG and the latency and the amplitude between the groups. And what we found in adults is that the migraine patients because I didn't explain that, but we also divided the migraine patients into two groups, so patients with aura and patients without aura. And we found that the patients with aura, they have higher amplitudes of CO2 response than patients without aura. So it seems that their impairment in pain processing is bigger than for the patients without aura. So even the brain processing of pain stimulations in the brain is also different between these two groups of patients. That's what we are doing.
Yeah, exactly. And as Dr. Mignot explained, it's very important to design your study in a way that you are getting the results that you're getting. And actually, the whole point of this lecture or this webinar is basically that you should be able to see if any study that you're potentially thinking about, because you might have some ideas about some research projects, but in that, you need to consider the design of the study very carefully. I myself started working with EEGs about a year ago, and I honestly didn't know much. I knew less than this. And it was difficult for me because I was not exactly able to design a study as I wanted, because there are so many things to consider. Like, for example, a student approached me with saying that I wanted to design a study where I record people doing some activity and I want to know what their brain does while doing that activity. Now, the problem is that you can't really use an EEG method for doing a study which involves a lot of body movement, which might involve sweat. Because the thing is that then you will get so many artifacts that you might not get the power of research that you are looking for. So that's why the whole point of this is that you should be very aware of all these things when you are designing studies. So we don't have any more questions. So I think we can move on to the fMRI part of the webinar.
Good, let's do that. Yes, so just to finish on that, as you said, it's important that you have this in mind. And I think the key word here is artifacts. Like, so you know when you're reading also an article with EEG, you know why there are all these pre-processing steps of filtering and so on. It's to get rid of these artifacts. So yes, you'll see that it's a little bit the same with fMRI data actually.
Excuse me.
Yes, sure.
Yeah, I wanted to know, like, with respect to the subjects in the before experiment, did you find the amplitudes being different for different subjects? Like, are the response strength of the response is different for everyone?
So yes, they are different. You mean in healthy subjects, for example?
Yes, yes.
Yes, so they are different because it depends on like the brain of each person, but usually it's not that the standard deviation is not so high usually. So you can easily compare two groups of people, but it's never exactly the same number, the same amplitude. But the shape should be the same and the latency is usually in a certain range. So when I said it should be the same amplitude between subjects, it's not true. It's just that there's a range that is considered to be good for healthy subjects, for example. Was it clear, the response?
Sorry,
Do
thank
you
you.
want me to explain in a different way?
No, no, no. Thank you so much.
Good. So then I'll continue with, if there's no other questions, I'll continue with fMRI and MRI. So yes, we'll go through all these steps, magnetic resonance imaging, Bolt signal, limitations, developing a paradigm, and what do you do when you have images in the end. So why using MRI and fMRI? So first, I didn't say that, but when you use EEG, you have a very good temporal resolution because you're able to acquire data every milliseconds, which is not the case with MRI and fMRI. But with EEG, because the electrodes are placed on the scalp, you can't really know from where the signal comes from. So that's why MRI and fMRI are very good, and they have very good spatial resolution. And they are, as well as EEG, they are non-invasive techniques. And the MRI and fMRI allow investigating anatomy and cognitive processing at the same time. So it's very convenient. When I said that there's a good spatial resolution, it's because you can acquire, so voxels, because we don't talk about pixels as it's in 3D, but we talk about voxels. You can acquire voxels of less than 1 cubic millimeter. So it's very good. And I will talk about the different physiological mechanism, because MRI, so for the structure, and fMRI for the functions, lay on different physiological mechanisms. So I just give you an example of MRI study, what you can do with some images of MRI. So you have the original structural image. You can remove everything that is not brain related, and then you can determine the borders of the cortical gray matter, and you can measure the thickness of this cortical gray matter, and you can do that, for example, to compare two groups of subjects, so healthy subjects and Alzheimer subjects or whatever. This is an example of what you can do with MRI data, structural MRI data. With fMRI, you can see the activity of some areas in the brain. So it's more related to the function. So for example, here on the top, you have healthy subjects, and on the bottom, you have stroke patients. And so in this study, they asked people to try to do a hand movement, and they looked at their activations in the brain with fMRI in contralateral hemisphere or ipsilateral hemisphere. And they compared these two groups of patients, people, and they showed that for the contralateral hemisphere, you have lower activations for the stroke patients, but you have more activations for them on the ipsilateral side. So this is just an example of what you can do with fMRI, but we'll go back to that later. So I am going to explain the very basic principle of magnetic resonance imaging. So as I said in introduction, it's very simplified. So if you want more information, you can refer to books or websites that are more specialized on that. So as you all know, human body consists mainly of water, and water is oxygen and 2 hydrogen atoms. And concerning the usual MRI, it's the hydrogen that is imaged. You can image other types of atoms, but they need to have a specific magnetic property. And we are not going to talk about it today, because concerning the brain imaging, it's always mostly hydrogen. So the hydrogen nucleus, H+, that we can also call proton, can be considered as a little magnetic dipole. It means that when it's placed in a magnetic field, it acts like a little magnet, and it can be tilted with the magnetic field. So what happens in an MRI machine is that we apply a magnetic field, a constant magnetic field. So all the hydrogen protons in the body are in the same basal state. And then we send a radio frequency pulse at a certain frequency, and it involves a transitory change in the proton's configuration. Okay, when you stop the radio frequency pulse, there's a recovery of this configuration of the protons, and when the protons is recovering, there's a sudden release of energy, and this release of energy is recorded by an antenna, and it's the core of the MRI signal, the release of energy from the proton. So if you code the magnetic field, so you apply a constant magnetic field, so all the protons are tilted in a certain way, but so if you do that, you don't know exactly how to select a certain slice in the brain to image it. So you need a gradient. You don't need just a constant magnetic field. So in addition to the main magnetic field, there are some parts of the MRI called gradient coils that allows you to add to that a little gradient of magnetic field. So in the three dimensions, so there's 3 gradient coils, X, Y, Z, and these 3 gradients, they allow you to select in space where are the slices in the brain that you want to image one by one. Then you can reconstruct a 3D image, and this 3D image is called a volume, using an algorithm that is called Fourier transform. And I told you that when you stop the radio frequency pulse, then there's a sudden release of energy. And this sudden release of energy in each voxel is translated in a color on a gray scale. So the protons are not in the same amount everywhere in the brain, because depending on the tissue, you don't have the same amount of protons. So there's not exactly the same amount of energy that is released according to the placement in the brain. So that's why you can have a translation in a color scale, and that gives you this kind of image. So when you look at the MRI machine, you have this tunnel here that contains the magnet and the gradient coils. So the main magnet for the main magnetic field, and the gradient coils for coding in space the magnetic field. And here, around the head of the subject, you have the transceiver or antenna, which is responsible for sending the radio frequency pulses, but it's also responsible for recording the release of energy, so the signal, the MRI signal. So the signal intensity at one location depends on the proton density in a specific tissue, but it depends also on the parameters of the pulses, the number of pulses, and the time between 2 pulses. So this is a bit complicated. I don't want to go into details for that, but all these parameters, they are usually called the sequence. And when you read an article with MRI, you will see that there are some different type of contrasts, and this contrast depends on the sequence that was used. So that's why you can read T1 weighted or T2 weighted contrast or proton density contrast. It depends on the parameter that were used. And that's why sometimes in some contrast you have the gray matter is black and the white matter is white, and vice versa. But yes, the signal intensity is also depending on the blood flow. We'll see that with the fMRI later. And it depends also of the use of a contrast medium. So if you want to increase the signal in your image, you can add a contrast. Sometimes they use gadolinium or things like this. Okay, so if you read an article with MRI, I just want to give you some basic parameters so you can understand what it means. The TR is the time to repeat. So it's the time between two radio frequency pulses. So it's the time that takes the machine to acquire one volume. Then you have the TE, it's time to echo. It's the time between the radio frequency pulse and the recording of the signal. You can see also the voxel size, so it's the size in millimeter of one voxel in the image. And you have often also the field of view, so it's the size in millimeter or centimeter of the grid containing the volume that you imaged. So I hope this can be helpful for you if you have to read an article about it. So now we are moving on to the bold signal. I don't know if you had some questions about the MRI principle already. If you have some, please let me know.
There are no questions in the chat box, at least. But if there are any questions, we can wait for a minute.
Yes, it's okay. If there are questions that comes later in the chat, let's see.
Okay.
So for the BOLD signal, BOLD is for blood oxygen level dependent. And it's based on the fact that when neurons are activated or when they are working, they need some glucose. And the glucose is degraded by neurons aerobically, which means that the neurons need some oxygen to degrade the glucose. This is possible thanks to the neurovascular coupling. So if you look at here on the bottom left, the neurovascular coupling allows the endothelium to be coupled to the neuron. And this is through this that the oxygen can be consumed. So when there's a brain activation, the neurons need oxygen to degrade glucose. So that's why there is an increase of the O2 consumption, which means that locally there will be a local concentration and an increase of the local concentration of deoxyhemoglobin, because it's the hemoglobin which carries the oxygen. So if it gives the oxygen to the neurons, then there's more deoxyhemoglobin than oxyhemoglobin. And then because there's this sudden O2 consumption, there's a need of O2 in this area. That's why there's a sudden increase of inflow of oxygenated blood and a sudden decrease in the local concentration of deoxyhemoglobin. So when you have this difference between local, a sudden increase of the inflow of oxygenated blood, it induces a local distortion in the signal, because it's changing the magnetic parameters locally. And this is the distortion that we can measure, and that is representing the BOLD signal. So the increase of the MRI signal locally, this is what we measure as the BOLD signal. And it gives an information about what kind of neurons and where the neurons are activated. But if you look at the response here, so it's the modelized BOLD response, it's a very long response. So when you have a stimulation, you need at least 7 seconds before having the peak of your response. And then you need more seconds so before your response fades away. So that's why we say usually that the fMRI has a very good spatial resolution, but a very bad temporal resolution, because when you send a stimulation, you need to wait a long time before seeing the response in the brain. So as I just said, the limitations are poor temporal resolution. Also because the air cavities can induce some distortions in the signal. There are some sinuses in the frontal part and in temporal part that give some signal dropout. So that's what you see here on the image. You can have signal dropouts in prefrontal and parietal regions. Also, if you move in the scanner, then it induces some artifacts. You can see artifacts here, like there are some stripes in the image. So yes, these are the limitations of fMRI. So when you want to develop an fMRI paradigm, there are some questions that you need to ask yourself before beginning. First, the goal. What do you want to observe? So, for example, if you want to observe which brain regions are activated when a stimulus is presented, so you can decide if you want to see in if you have a priori or if you have no a priori on the brain regions that will be activated after this stimulation. So if you have no a priori, you can use whole brain analysis. But if you have an a priori, you should use region of interest. It's usually called ROI analysis. And in this case, the question is a specific region activated when a stimulus is presented? So in this case, you use some standardized templates to define your region of interest and to see if this region is activated when you send your stimulus or when your participant is doing a specific cognitive task. You can also have a question about the networks, the brain networks, so which regions work together in resting state or cognitive state, or when a stimulus is presented. So in this case, it can be with or without a priori, and it can be a ROI analysis or a whole brain analysis. So in the two first case, you measure just simple activity. You want to know if some areas are activated. In the last case, it's functional connectivity that you are doing. So you just want to know which regions work together for a specific process. So you're talking of brain networks. Second, after the goal, you should ask yourself also what kind of task do you want to use and which cognitive processes are involved. So first, do you want to use a task, a cognitive task, or do you want just to see what happens in the brain when nothing is happening, where there's no task? So it's resting state. It has been shown before that when you're doing nothing, you just lay in the scanner and you're waiting there. So it's called the resting state. You still have some activations in the brain, and there are some specific networks that are activated. So for example, the default mode network. So you need to know that before. Also, if you want to use a stimulation, what kind of stimulation? Do you have a priori on the areas that will be activated or not? So this is another question. And does it involve, so the task you want to use, does it involve several cognitive processes? So this is more about the task you're using. You can have very low level cognitive tasks. So for example, just passively looking at a screen with some images, or you can have a very high level cognitive task. So looking at pictures, but asking your participant if what they see is edible, for example. So here I just give you an example of, so it's not a real study, I just took the image from an existing study, but what I'm saying is not related to this study. So just imagine that you want to see if a particular odor is changing the way you're processing the valence of an image. So you can give some happy images to your patients, some neutral images or some negative images. And at the same time, you want to send an odor in the scanner to see if the brain processes to these images are changing. So I just gave you a photo of olfactometer. So this is the machine that we use to send odors sometimes in the scanner. So in this case, you have two different kinds of stimulation. You have the visual stimulation and you have the olfactory stimulation. So what you need to ask yourself is like, what are the cognitive processes involved, because you'll ask your participant to rate like the pleasantness of the images, for example, but is it important that the odor itself is neutral or positive or a negative odor? Should it be sent at the same time at the images or in between two images? So this is the kind of questions you have to ask yourself. So in this case, in my example, what odors, when, at which concentration, which timing, how, knowing that, as I said, the response of the brain to one type of stimulation, when the activation, when the neurons are activated, this response is very long. So if you use, if you have two cognitive processing that are going on at the same time in two different parts of the brain, you will have a very long time and you will not be able to see which cognitive processes belongs to which type of activation. Then you have also to decide the design. So do you want, there are two main types of design, the block design and the event-related design. So the block design is, for example, in this case, you have, concerning the images, you'll have happy, 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 neutral, 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 negative, negative, negative. The event-related design would be happy, happy, negative, negative, neutral, happy, negative, and so on. So it's a bit randomized. So you have to decide between these two blocks. There are some good and bad things with both of them, but it depends on which type of stimulation you're using and what is the timing of your stimulations, if you are risking to have habituation to the response from the subject or not. So it's a question you should ask yourself too. So be careful with stimulus duration also and interstimulus intervals, because as I just said, you can have habituation. So if your stimulations are too close to each other, then the brain response decreases with presentations. And you can have also fatigue from the participant in the scanner. And when the participant is bored, the quality of the signal is less. So keep the experiment as short as possible. And also another very important thing is the control. So what is your baseline? Because as I said, the bone signal is not an absolute activity, it's an indirect measurement. It's not the activity of the neuron itself, it's the sudden increase in blood flow locally. Plus, the resting state, as I said before, induces also activations. So if you want to know which areas are activated in response to certain stimulations, you need to compare this activity across different conditions, and you need to have a control to subtract. So there are two ways of analysis in fMRI. You can have a within subject level. So you have to imagine a control baseline at the level of your participant, or you have the across subjects level, where you have to imagine controls in the level of a group of people. So with the within subject, for example, you can have a cognitive task and a stimulus. So you should design your experiment in the way that you have some timing where there's no task and no stimulus, where there's the task but no stimulus, where both of them are present, and where you have just the stimulus and no task. So then, depending on your research hypothesis, you decide which of these three should be your control. On the group level, so you can have different ways of doing analysis. So you can be interested in knowing if the activations are the same between all people from the same group. You can also see if the activity of your area of interest correlates to some variable. Or you can decide that your control can be, for example, another group, in the sense that you have a patient group but you can also recruit healthy subjects as a baseline. So when you have an activation, it is always compared to another condition, as I said. So you don't say the insula is activated when the stimulus is presented. You say the insula is more activated in the presence of the stimulus than in its absence. So usually when you read an article, sometimes they just use the first type of sentence, but you have to understand that it means that they did that against a control baseline. So finally, when you have your images, so you did your paradigm, you acquired your images, you have to construct a pipeline to be able to remove the noise from your data, because the bold signal represents only 1% of the total signal in MRI. So it's not a lot. And during a cognitive task compared to a basal state, there's only a variation of the signal of 5 to 10%. So you need to get rid of a maximum of noise. And the noise can be distortions of the signal due to sinuses, movements, heartbeats, and so on. So you need some steps to correct the images before being able to analyze them. So it seems a bit complicated. I'm not going to explain it in detail. I'm just explaining the steps in the middle. So for example, you can take your images and start with realigning them. If your participant moved into the scanner, maybe one volume is not realigned with the next volume, so you need to do that. Then you can co-register your functional images on the anatomical MRI, so which means that you're able to see that this particular activation is in fact the insula, for example, of your patient. Then you need to segment your data. So if you want just to use the white matter, the gray matter, or the whole brain, but not the skull, things like this, so you can segment it. Then you need to normalize your data. So it means that now you have the data for one participant, but you want to see for the whole group what happens. So if you want to be able to compare the brain of one participant to the brain to all the other participants, you need to normalize your whole group to have like a standardized brain. So for that, you can use different standard templates that exist. The most famous one is MNI, but there's also Talerac and some others. This is for usually healthy subjects. If you're working with patients, you can also do your own template with your own group of patients to normalize all your subjects between each other. Then you can smooth the data. So it's just a process to average the data, the intensity of one voxel with the neighboring voxels. You average the intensity. And then you have your images. They are almost clean and you can analyze them. And again, I'm giving you just some few examples of analysis, but it's just names. I'm not going into detail. So with simple activation and deactivation, you can use general linear model. And with functional connectivity, when you want to see brain networks, which means that you want to see how some brain regions work together. So you can have all of these kind of analysis, seed-based analysis. So it's when you want to see if one voxel works together with other voxels. Roy to Roy analysis, when you want to see if one region works together with another region. Independent component analysis, so when you don't have any a priori on your data and you want to see what are the individual networks during your session. And dynamic causal modeling, so it's again something else about brain networks. So yes, the take-home message for both, actually, for EEG and fMRI, is that you need to think about your experiment before running it. it seems a lot like this, but it's a lot of fun to think about the whole process and about the whole designing the whole experiment, and then choosing the stimulus and the stimuli and the tasks, and then getting the images or the signals and filtering them and choose the pre-processing steps. That's true, it's a lot, because it's not like a black box where you put your data, you click on a button and you have the result in the end, but it's a lot of fun to do. So you have to decide beforehand if you have an a priori on the structures involved in the case of fMRI. If you want to include whole brain or only ROIs. In both EEG and fMRI, if it is a resting state or task-based paradigm, which stimuli you want to use, at which arousal, which balance, duration, is it okay with the context of your experiment? What is your control condition? And do you need a block or an event-related design? So yes, that was all for me. I hope you could find something to learn here. And if you have any questions about this part, don't hesitate.
Thank you for the presentation. I think honestly, it was a complete revision for me because I had also forgotten a lot of this. So it was quite useful. If anybody has any questions, you can ask them by turning on your mic, or you can ask them in the chat box. So, and if any of you want to ask her something else related to neuroscience, related to neuroimaging, how she got into it, I'm sure she will be happy to answer it.
Yes, of course.
Yeah. And if you have any questions for me as well, how I got into like EEG and how my future project is going to be MRI based after doing an MBBS. So you can ask me that as well if there are any questions.
If not, it's also fine.
Yeah.
I just hope that it was not too like that you could see the use of it for yourself.
Yeah, true. Actually, some of them are asking, we'd love to know more about your journeys. And I am sure that, so this is more of a casual discussion, really.
Yeah, no problem.
Nothing
It's okay.
related to anything educational, but I think I know that for Dr. Mignot, it has been a very roller coaster ride where some projects were great. In some projects, they weren't so great because she is great at what she does. Yeah, that's true. But the supervisor matters a lot. The lab matters a lot.
That's
and I
true.
know that she's happy over here.
Oh yeah, I'm very happy in Dresden. Yes, that's true. No, that's true. Actually, I really liked at least to have the opportunity to do EEG and fMRI, not at the same time, of course, but I think it's kind of complementary. So that was a very nice thing. Yeah, that's true. And here now I'm doing mostly EEG, fMRI. I don't do it anymore, but for sure in the future I probably have some studies about it.
And your previous, like your PhD was based on fMRI because it was,
yes.
Yeah. And yes, I actually wanted to ask you that from your undergraduate in cell biology and biochemistry and everything, how did you decide to get into neuroscience? Like what made you decide neuroscience?
That's a very good question. So I'm not sure it's very professional, the answer you will get. So I started to study biochemistry and genetics. And I had just few neuroscience courses during this bachelor degree. And basically I hated biochemistry. So
as we all do. I think you guys can write in the chat how much on a scale of 1 to
yes,
10 you hate biochemistry, because
you can
everybody hates biochemistry.
please use the thumb in the reaction if you hate biochemistry. But I really hated it, and I was very interested in everything that could be at the border between at the border between mind and psychological things, but also a way to investigate it with biological substrate. So that's how I got to know EEG when I was in master's degree.
Yeah.
And it was very interesting, even if I was a bit afraid at first with all, because it's a bit an engineer thing, like a nerd thing. So I was a bit like, okay, I like psychology, I don't know why I'm gonna work with EEG, but I like it because it's very pluridisciplinary thing, because you have this time where you're focused on the pre-processing steps, and it's a computer thing, and you have these steps where you have to think about the experiment, think about what would the patient think, and then think about the interpretations in a psychological way and so on. So yeah, that's why I like this.
Yeah, and it's very, very interdisciplinary, as you said. And I think, like you said, for doctors, like the whole clinical aspect of it, like, because even if you don't enter the field of neurology, you are going to do an EEG at some point in your life. You are going to come across a brain MRI at some point in your life. And just knowing what goes on behind the scenes is what happens in research, basically. Like, if years from now, we are using EEG as a diagnostic tool for migraine, or if we are using like the size of the olfactory bulb as a diagnostic or prognostic tool in many diseases, which actually it is being used right now, then it'll all be because of research. And I think that's why especially neuroscience is so beautiful, because the brain is just endless. And I think even Dr. Mignot can agree that every day we learn new things. Like with every experiment we do, and I at least have only done like 2 studies related to EEG in the lab, but she has been working with EEG and MRI for a very long time. And I hope that we, especially her, she was able to contribute to your knowledge of neuroscience and neuroimaging, and that you keep all this in mind the next time you try to design a neuroimaging study, or any study for that matter, and that you can show off a little to your friends as well.
Exactly. I know it was not 100% clinical. I am aware of that, but I hope you could get the message. So thank you very much.
No, I think it was very good to hear it from like the horse's mouth, like how everything happens, because clinicians are always focused on, oh, this disease, oh, that disease, but no one really talks about what goes on behind the scenes. So yeah, it was very good to know. Thank you very much.
Thank you.
And yes, thank you everybody for attending. I will be putting up the video for this on YouTube as well on our channel, and I'll be uploading it today, because the video from last time, I still haven't uploaded it. Sorry, I got busy during the week. But this time I'm going to upload it today itself, especially since it's a bit shorter than last time, because last time it was like almost 2 hours. So yeah, I'm going to upload it. Does anybody have any closing questions or answers?
Yeah. Hi.
Yeah.
I actually wanted to ask. So, like, in India, like for these research work, you need a lot of equipment, or I'd say a lot of resources.
Yeah.
How do we go about it? Like, if I want to, do anything with respect to EJ or imaging,
Yeah.
how can I go about it? Like, I'm not sure if, uh, I mean, you guys are aware of it or not, but I just,
Yeah, yeah, of course. I don't know where you went. Are you, yeah, you're still here. Yeah, so I think as far as I know, um, EEG and MRI is actually available in all hospitals. I mean, I studied in a semi-rural hospital, and we had like EEG facilities and MRI facilities, and that is a start, of course, because you might not have like the equipment for providing the stimulations, etc. But you can always do something that you're doing on your own. Like, for example, where you're not using a machine, but where you're using something as simple as a screen in front of the patient, and you're showing them certain images or you're giving them certain stimulations. It can be something as simple as playing a sort of music or doing something which is a stimulation of any sense. And then you can record an EEG, and that is very, very possible. You don't really need any sort of high end equipment for that. You just need plain EEG material, and that is available because a lot of diseases use EEG as a diagnostic tool, be it epilepsy or be it any sort of electrical disturbance. Even in something as basic as any neurological deficiency, an EEG is always done. Even in the case of, for example, if there is loss of consciousness and there is lack of recovery of consciousness, then also an EEG is done. So EEG is very commonly used. I know that. So you can definitely do it. When it comes to MRI, it's a bit more complicated because you need the stimulation to happen while they are in the MRI machine. So that can be a bit complicated because you need a screen which is like put up inside the MRI machine to sort of stimulate you, or you need a pipeline which is coming from outside, which we use actually, which is sort of going into your nose and giving you stimulations related to olfaction. But yes, if you want to do an EEG study, it's definitely easier in India. If you want to do an MRI study, it is of course a bit more resource heavy, but EEG is definitely doable. I will say that.
And I think also that the EEG, it's at least like this in Europe, so I guess it's the same in India, but it's definitely cheaper to do an EEG than an MRI scan.
Yeah, that is true in India as well.
And the also good thing with EEG is that you can carry it to the patient's bed if needed.
Yeah, it can be done bedside as well. That's also like a good point. Because sometimes it's not possible to mobilize the patient. And for example, if just off the top of my head, if you choose a neurological disease, right, in which patients are hospitalized for the neurological disease, then in that case, you would have to use a bedside EEG, preferably. And you can see, like, so you can take a bunch of healthy subjects, participants, and you can choose these patients, and depending on how many stimulations you give them, you can increase or decrease the sample size. Dr. Minyo and I were discussing this the other day, that if the number of stimulations is a lot, then the number of people required, that is the number of participants, the sample size reduces. Of course, it is not preferable to go below 10 because then it becomes like a pilot study. But anywhere between 10 to 30 can be done if you increase the number of stimulations. That is more than, say, 40, I think. Like, because I had 20 stimulations in my study, and we can't do less than 10 participants.
Yeah.
Yeah.
The number of stimulation depends also on which type of stimulation you're using. For example, CO2, it's a very clear response. You don't need a lot of stimulations to average. But olfaction, for example, is a very weak response. So you need more. And yes. So, and also knowing that when as I said, the longer the experiment is, and then the subject gets tired and the EEG is not a good quality anymore. So that's a compromise to find.
Yeah, that's true. And as she's saying, related to olfaction, we were also doing a study related to virtual reality, that is VR, which is all the craze, has been all the craze since I think 2012 or 13. But we were doing that in which we were showing participants the like a VR image, like a video was going on, and then at certain points we were seeing if there were any potentials that were evoked. And in this case, it wasn't an event related potential, which was exactly what Dr. Mignon was saying, that there can be two types of potential. There can be evoked potential and event related potential. But yes, it can vary according to the experiment itself. Yes. Yeah, so any other questions? I hope that answered your question, by the way. I know it was a very long
Yeah. Thank you so much. No problem.
Okay, great, perfect. So thank you, everybody. Thank you, Dr. Mignon, for taking out the time. And thank you for presenting this amazing presentation to us. It was very well made. I really enjoyed it. And I think so did everybody else. So yeah, I'll see you guys tomorrow if you guys are going to come for tomorrow's cardiothoracic imaging, not imaging, cardiothoracic tools webinar. And yeah, see you tomorrow. And Dr. Mignon, see you on Monday in the lab.
Thank you very much. Thank you for inviting me, and thank you to the people who do not like biochemistry.
Yeah, I think that's all of Indian medical students.
Okay, thank you.
See you. Bye-bye, everybody.
Bye.